My name is CHE120N, and this is the science behind COVID-19 testing. The coronavirus has changed our lives dramatically. It has flipped the world upside down and has transformed the way we live today. It can't be denied that the coronavirus has had a negative impact on the year 2020. Nevertheless, the science behind COVID-19 testing is fascinating. So in my presentation, I'll be explaining what the coronavirus is and the different types of tests that are currently available. I will also be describing the science behind each one of those tests. What is the coronavirus? The coronavirus, according to Johns Hopkins Medicine, is a respiratory disease that is caused by a specific virus called SARS-CoV-2. And that virus causes a wide range of different symptoms, such as fever, cough, shortness of breath, and other flu-like symptoms. The first test I'll be explaining is a diagnostic test called polymerase chain reaction, or PCR. And this test is actually a very common technique used today because scientists are able to take a small sample of DNA and create millions of copies of that DNA so they can analyze it and determine if one has the virus or not. Before I explain the PCR process, I just want to go over some unfamiliar terminology. So PCR stands for polymerase chain reaction. RNA is a sequence of transcribed DNA that can carry genetic information for many other different viruses, not just specifically for COVID. cDNA, or complementary DNA, is a single-stranded DNA sequence that has been synthesized from an RNA strand. And then primers are just small DNA sequences, and then probes are DNA sequences that can detect different sequences. So the first test I will be describing is the PCR test, and according to Tanner S. from discovery@mayo.edu, the test first starts out by taking a nose swab to obtain some DNA from the patient, and then this nose swab gets placed into a test tube that contains a buffer solution, and this test tube is then sent to the lab where they are able to extract only the viral RNA. And then the scientists are able to add an enzyme called reverse transcriptase, and this enzyme is able to convert the RNA into complementary DNA, and this complementary DNA is used as like a blueprint for the rest of the process. And then the tube gets placed into a thermal cycler, which is a machine. It's a machine able to regulate the different temperatures throughout the whole process. So the first step in the PCR process is called denaturing, and this is simply where the strand is broken up into two single strands of DNA. And this is done at usually a higher temperature because the hydrogen bonds between the strand need to break apart and create the two strands. The second step in the process is called annealing or attaching, and this is where the primers are going to attach to both sides of the DNA strand, and then the probes are also going to be introduced at this point, and they're going to attach by binding onto the small specific sequences present only on the viral cDNA. The last step in the process is called extending, and this is where a new enzyme is introduced called DNA polymerase, and they're going to start on the primers and replicate the sequence. And as it's replicating, it's going to hit the probes, releasing a fluorescent molecule. And this process will continue until millions of copies of DNA sequences are produced. And based on the fluorescent molecules and how the intensity of light is produced, it can determine the results on the test. So a high intensity of fluorescence is considered positive, and if there are little to no intensity, the test is considered negative. The next type of test I'll be explaining is the antibody test, and there are two different types of tests that are currently available. The first test is using a technique called enzyme-linked immunosorbent assay, or the ELISA test, and the second test is using it's called a rapid antibody test using a test strip. So the first test I'll be describing is the ELISA test. And according to fredhutch.org, the process first starts out by taking a blood sample, and this blood sample gets placed into a machine called a centrifuge, and the centrifuge is able to spin very fast, eliminating all the red blood cells and leaving behind only a serum that contains any antibodies present. And then dishes are pre-prepared before the test, and these dishes contain live SARS-CoV-2 antibodies and a fluorescent enzyme as well. So once they have the serum is placed onto these plates, and the antibodies will start to bind to the proteins, the live SARS-CoV-2 proteins, and produce a glow. And based on the glow, it can determine if one has specific antibodies or not. The last test I'll be explaining is the rapid antibody test using a test strip. And according to researchamerica.org, this test first starts out by taking a small sample of blood, placing it into a well that contains a buffer solution. And the solution will start to travel down towards the test strip, and it will hit the first pad called the conjugate pad. And this pad contains live SARS-CoV-2 proteins, and if the patient contains any antibodies, it will continue down the strip, hitting the IgM, or test line 1, that contains IgM antibodies. And IgM antibodies are antibodies that appear quickly after the infection, and if they contain any more antibodies, it will hit the test line 2, which contains IgG antibodies. And IgG antibodies are antibodies that remain in your blood for long-term protection. And then this is a picture of the different types of results one could get. So, and in this case, the control line is basically just a line that indicates whether or not the test was valid. And in conclusion, using these tests and the ones currently being developed today, we could possibly be on the other side of this pandemic. And here are my resources.